Hello, good afternoon. I hope you're all having a great symposium. I'm very excited to talk to you about deep sea jellyfish and the results of my previous postdoc at Jamstack with Google. They're three of the five co-authors of the results I will present today here in person, which is really nice after a long pandemic. So there's Dougal, Mayhill, and Alan Collins. So although this is a jellyfish conference, we barely talked about the midwater. I was really happy to see Atola this morning, but actually the midwater is the largest biome ecosystem on this planet, and it's also dominated by gelatinous zooplankton. It provides many ecosystem services like fisheries or the carbon pump. But the reason why we didn't see so much deep sea talks is not surprising, because there is a lot of difficulties that comes with sampling these enormous ecosystems. So we're not only speaking in terms of volumes, but also huge pressures. As a result, it's also very expensive to sample. And in terms of analysis, you really have to think in 3 dimensions. So can we therefore do more than just descriptive research? Can we do more than just say, look, we saw this nice jellyfish, it was in there? The answer is no, so thank you for your attention. No. No, there is many ways, and today I will talk about ecological niche modeling, but if you're interested in more genetics techniques, come to my talk. So what's ecological niche modeling? So this was a technique developed mostly first to study terrestrial environment. And what it is you collect some presence data, so latitude and longitude data of your species. You combine it with environmental maps that you're interested in, and then a model will calculate the ecological niche of your species, so which temperatures they prefer, and then we can project it again on the maps to find locations where there's a high probability to find your species. Now, about 10 years already, since thanks to Bastian Bentlagen, we are able to model also in 3 dimensions in the oceans, which is unfortunately not yet much applied. So I'm here to say, please model in 3 dimensions. And this is possible thanks to maps on environmental conditions that are now available in 3 dimensions. I use the one from World Ocean Atlas, but there are much many out there. So you basically decomposed in these different layers, but you still model in one big model, and then you redecompose afterwards your maps. And then in terms of presence data, you need also data in 3D, so latitude, longitude, and depth. Today, I will present a case study about the narcomedusa genus Solmissus. Why Solmissus? Because it's widespread, but since it's a narcomedusa, it is also feeding on other gelatinous zooplankton. Therefore, it has an important trophic role, as it's a top-down regulators of the midwater gelatinous web. Okay, so what happens when you start your postdoc on deep sea jellyfish in January 2020? This soon becomes your office, and now you have to find data for your postdoc. Where do you go? So luckily, there are some biodiversity databases out there, such as OBIS or GBIF. If you're lucky, there will be information on depth, which you need for your model, but none of them will come with a picture, so you cannot quality check at all. When it comes to deep sea imagery and videography, luckily there's much more databases out there, such as Jamstack or Embarra's PhantomNet, SeaTube, and more now of this expensive deep sea expedition will broadcast live their RV videos. So it makes it much more accessible to everyone to get this data. So my first step was to check which kind of data to use in my models. So I compared, on the left you can see a map using data from OBIS, with just the green points were the ones I had depth information that I used. And they are much more widespread than the ones I got from the deep sea databases that I could vet it, so which means I personally checked every image to make sure it's a Solmissus. And there, over 90% of the data comes from North Pacific, because institutes like Jamstec and Mbari are studying the midwater there, but other locations will focus mostly on the benthic and forget there is a whole ecosystem on top. So I modeled then the genus, and so on the left for obis, on the right for the Veta data, and in 3D, so we went from 200, 400. Warmer colors means higher probabilities to find Solmissus. And what you may have noticed is, so the colors or the distribution that was projected reflects the data that we put in, but how did these models perform? This can also be calculated thanks to a statistic called the continuous Boyce index, which goes to -1 to +1, where +1 is your perfect model. Will always find your jellyfish wherever it is, and like around 0, it's not better than random, where you just have thrown some points on the map would be equally good. And as a rule of thumb, if you have a CBI that's higher than 0.7, you have a highly performing model. And so for the ones with obese, we had an average performing model, but we had a very good score for the vetted data set. So here we can clearly see garbage in reflects garbage out, even though, well, I hope we'll have more data from other locations as well for the vetted data sets in the future. Okay, a bit more about the genus Solmissus. Oh, the time is going fast. Okay, so let's see. Say there were 3 accepted species for now. Before we started, included Solmissus incisa was a widely distributed species, and it was suspected to be a cryptic species. And with preliminary data in 16S, we confirmed this. And one of these cryptic species is likely Solmissus plicae, which has more than 28 tentacles. And when we look at where these are distributed, so on the right were the 28 and plus species, and on the left, we could see there was a division along the depth gradient, with the ones with less than 28 tentacles around 400 meters, and the other one around 800 meters. And this was also reflected in our model. So the way that we looked in 3D, we were able to also see there was some different niches, although there is some overlap in 2 dimensions. Now, very important when we are interested in climate change is looking at what are the environmental factors that are limiting the distribution of deep sea jellyfish. And here all our models, whether on genus level or on morphotype level, found that dissolved oxygen was the most important limiting factor, which you may find surprises. But when we looked, this really low concentrations is where you found the most of our jellyfish. And because I had checked all these pictures, I could also work on the reproduction of narcomedusa, which doesn't have a polyp benthic stage. It will produce jellyfish that will be produced by these oocytes. And we found there was no seasonality. We found that polyps with these visible oocytes all year round, and reproducing jellyfish were found in slightly warmer waters. So that bears the question, will these deep sea jellyfish be a climate change winner, as with slightly warmer temperature or very low dissolved oxygen, they're doing very well. So what can we conclude? I think I cannot stress enough the importance of online databases when it comes to do deep sea research when dealing with such a huge ecosystem. But please include some images or videos that we can check the quality of your data. Then we found that Solmissus incisa is likely a complex of cryptic species, and that there is some differentiation along the 3D axis, and that Solmissus was reproducing all year round, and likely with the upcoming climate change, it will be favorable for Solmissus, at least just for environmental parameters. We didn't look at any biotic interactions. So thank you for your talk, and if you want more details, you can check the papers or just ask me. Many thanks.